So good morning, and first of all, I'd like to thank Rajiv for a most generous invitation. It's wonderful to be here, and also thanks for putting together a fantastic program for this Congress. What I would like to do is, over the next 25 minutes or so, is to give you a bit of an overview of the work that we're doing in Victoria and Australia on molecular breeding and forages and some of the underpinning biology. So without any doubt, over the last decade or 2 decades or so, we've seen very significant advances in technology-enabled applications in systems biology. By systems biology, I mean being able to describe biological systems across the different phenomena and in an integrative manner from genome to phenome. The second, I would think, key point that I would like to make is one about considering the totality of the biological system that we deal with in crop improvement, and also to do so with a through value chain impact perspective. And specifically today, I will be talking about our work on the one side, better understanding a plant symbiome, and I referred specifically in that manner, not only considering the core, say, genome of the target crop. In our case, is perennial ryegrass. Perennial ryegrass accounts for around 70% of the total forage grass seed production for temperate agriculture worldwide, but also the supplementary genomes, the associated biota that we ultimately deploy on farm, and the implications of that for key traits such as biomass yield, forage quality, yield over time, so persistence, as well as aspects of both biotic and abiotic stress tolerances. So I've divided the talk into two components. On the one side, reflecting on some of the knowledge that we've gained over the last few years in systems biology of perennial ryegrass symbiomes, so with grass and its endophyte associations, as well as the forage grass associated microbiome. And specifically, we'd like to introduce, rather than the target species, the target plant, namely perennial ryegrass, some of the endosymbionts that play a key role in ultimately pasture performance. And these are fungal endophytes of the genus Neotyphodium. These are asexual filamentous fungi, and they form mutualistic symbiosis with temperate grasses. They are strictly seed transmissible, so form part of the genetics that we deliver on farm when we sell perennial ryegrass seed. And the endophyte benefits from the association by being protected by the host, and as well as receiving nutrients. And importantly, these fungal endophytes protect the grass, the host grass, from both biotic and abiotic stresses. And part of the mechanism that they use to do so is to produce several bioactive secondary metabolites that do so in plant. I will tell a little bit about more of that later. And these fungal endophytes establish in a sense, species-specific associations. So in the case of perennial ryegrass, Lolum perenni, there are basically 4 different taxonomic groups, and one of those is Neotyphodium lolli, derived from the grass choke epicloid pathogen from Epicloid festucae through loss of the sexual state, and other taxonomic groups such as LPTG2 that are generated through interspecific hybridization between Neotyphodium lolli and Epicloid typhina. And here you see also some of the bioactive molecules, and some of them are beneficial in a sense for pasture performance, since they will confer resistance, say, to insect pests. But others obviously have a anti-mammalian herbivory activity, such as Lolitrum B, and part also gavalin, and are undesirable. So the way that we approach that is considering, in a sense, the symbionts as part of the germplasm collections that we want to deploy in the genetic improvement process, and starting from a global endophyte discovery, and then characterizing that through to pangenome analysis. And the example I'm illustrating here is the one of, in a sense, exploiting global genetic diversity of fungal endophytes of perennial ryegrass. The way that we deal with that is on the one side with identified from genetic resources and seed collections of the host. We look at using molecular markers to detect the presence of the fungal endophytes, and what we find is that genetically similar endophytes will also have a similar bioactivity profile and origin. So here you see examples of endophytes that will produce, say, pyramid only or pyramid in origin B. So pyramid is a key insect deterrent, so one desirable alkaloid. Another major cluster in Neotyphodium loli producing origin B, ergovalin and pyramid, and so on. So we find also that some endophytes will show reduced toxicity effects. It means that they will not produce the anti-herbivory alkaloids that have anti-mammalian activity. We find also that we can exploit the correlation of genetic diversity with geographic origin and toxin profile in order to be able to discover desirable fungal endophytes for the deployment in the genetic improvement process more effectively. So we could, based on the genetic profile, predict the bioactivity profile of the endophyte and as well as informing new germplasm collections. So in a sense, it's a broad applicable approach for the discovery of novel endophytes, and we have used that not only in perennial ryegrass but also in tall fescue as well as in warm season grasses such as brachiaria. And in that manner, we built up a collection of a broad-based germplasm collection of novel and genetically diverse endophytes, and here are some examples of those belonging to different taxonomic groups: Neotyphodium loli, the endophytes of perennial ryegrass, Neotyphodium xenophyalum, endophyte of tall fescue, and a range of other taxonomic groups of fungal endophytes that will colonize both perennial ryegrass and tall fescue. Established those in vitro cultures, and we confirm the genotypes of the endophytes, and we can also cryopreserve them and for long-term deployment. The other approach, or the complementary approach that we use, is then selecting across the diversity panel of endophytes representative across the different groups, and subjecting those to a pangenome analysis, and in the manner characterizing both the core as well as the flexible genome. Establish a reference genome for Neotyphodium loli, and we have now sequenced over 80 ryegrass endophyte strains, and covering endophytes that are in commercial deployment today, others that are in pre-commercial development, as well as a global diversity across the different geographic origins of these fungal endophytes. So a pangenome analysis across spectrum of genetic, geographic, as well as taxonomic diversity, and this provides us with very useful information in order to be able to select endophytes for and confirm also their desirable spectrum of bioactivity. So what you see here is three different taxonomic groups of fungal endophytes that colonize perennial ryegrass, the major one, Neotyphodium loli, LPTG2, and this one, which we call now LPTG3. And here you see all the genes involved in the biosynthesis of the major bioactivities: pyramine, the insect deterrent, ergoalkaloids that also confer part of insect deterrents, and lolidrums B, that are the ones that cause a ryegrass stegose. And it's with all the genes involved in the biosynthesis of these compounds, and you will see that in this manner we can find presence or absence of those and identify fungal endophytes that will produce the desirable alkaloids, such as, say, the production of pyramine, but they will lack genes such as the SILT-M and J genes, the two critical genes in the production of lolidrum. So they will be, in a sense, safe for deployment and in a genetic improvement process that will be animal safe, but at the same time confer the desirable attributes of biotic stress tolerance. Now, how can we characterize then these endophytes over time in a way that, and also dissecting the effect of the genotype of the endophyte and the effect of the genotype of the host plant? And the way that we do that is we establish a diverse panel that is of isogenic hosts from a range of varieties of cultivars that correspond to very different types of the host of perennial ryegrass, and we can immortalize that individual genotypes of the host so that we can then inoculate them over time with a range of endophytes and compare them side by side. So a very powerful research material for doing a comprehensive symbiome analysis, and in that manner being able to dissect the effects of the genotype of the endophyte times the endophyte, the genotype of the plant. So this illustrates this step in that process, and we confirm then, in a sense, having established then that panel, a matrix of grass genotypes, host genotypes, and endophyte genotypes, and being in that manner able to dissect not only the effect of the interaction of the genome of the host with the supplementary genomes of the fungal endophytes. So, starting from the genome, now moving into what we can learn out in using these materials in the context of the interplay. There's a host genome and supplementary genomes at the transcriptome level. So what we'll see here is, so we've done a RNA-seq libraries both from shoots and from roots of perennial ryegrass, single host genotype with in presence or absence of a fungal endophyte, and grown in different nutritional conditions, and now asking the question about what are the transcripts of from endophyte-coded genes. And so here you see the libraries right into shoots, right into roots, and you see that the endophyte transcriptome is exclusively present in the shoot of the symbiotum. In a sense, the endophyte will colonize the aerial part of the plant. Now this looking at the reverse situation, what's the impact of the endophyte, so endophyte-regulated plant genes, so looking at both the shoot transcriptome and the root transcriptome from a perspective of the grass host. And what we see is that we have around a bit over 900 endophyte-regulated plant genes in the shoot transcriptome, and with 68 of those that are differentially regulated and in shoots. And now looking at here the reverse situation, what's the impact of the supplementary genome of the endophyte on the root transcriptome, and we see that the majority, so actually over 720 plant genes that are expressed in the roots are differentially regulated by the presence of the endophyte. So doing a genetic cluster analysis, we find 13 different, in a sense, patterns of expression. And I will show you only one or two examples of that. In this particular case, is a cluster of genes that are, so these are root expressed plant genes that are induced by the endophytes. So you see the samples from shoots and from shoots and roots, in absence of the endophyte and in presence of the endophyte. So the more yellow, the more, say, upregulated it is. So this actually represents the largest cluster of endophyte-regulated plant genes. And if we look at the annotations of these plant genes that are regulated by the presence of the endophyte, we see that most of them correspond to defense response genes. So in summary, I think the key point I'm trying to make is that we see that the endophyte will colonize the aerial part of the plant and will hijack the plant transcriptome machinery in order to protect particularly the root system that is not colonizing directly. So the symbiotum transcriptional response then to the endophyte presence is primarily an upregulation of defense-related genes in the roots. To give another example now, and this is another particular focus on the cluster number one. This is a range of root-expressed genes, and you see that they're particularly expressed in the root samples that are now repressed by the presence of the fungal endophytes. And if we look at the notation of some of those, particularly here's a nice example. This is MAX2F box leucine-rich repeat gene that is involved in signaling in cyclolactones. And if we look at the ortholock in Arabidopsis, we know that the gene, when there is a loss of function of that, leads to an hyperbranching in both in shoots and in roots. So, in a sense, not only protecting the root system by upregulating the phosphorylated plant genes, but also playing an important role in plant architecture, development and growth. So shifting now from, trying to take you through a journey from a global discovery of fungal endophytes, the pangenome analysis of those, the impact in the context of the transcriptome, of the association, the interaction of the host genome with its supplementary genomes, and now moving into the metabolome analysis side. So what you see here is a range of some of the core bioactivities, so a range of different endophytes. The endophyte toxin reduced at the level of the major components, so lolitrim, cygovalin, and pyramine. Well, we could compare some of the origin of those and some of the taxonomic groups with they correspond. So we do a comprehensive metabolome analysis, and we complement that with targeted metabolic profiling, particularly related to the production of the bioactivities of the alkaloids. So metabolic profiling across the spectrum of the genetic, geographic, as well as taxonomic diversity of fungal endophytes that colonize, that form associations with perennial ryegrass. Now, once we take these endophytes, we inoculate them or we deploy them in a genetic improvement process, what we see is that there are very significant differences and quantitative differences in the levels of production of the different alkaloids. So these are a range of different endophytes, the standard toxic endophytes. This is a Neotyphodium loli, LPTG2 and LPTG3. So in a sense, 4 different taxonomic groups of fungal endophytes that colonize perennial ryegrass, and seeing some of the key bioactive compounds, xanthitrim, pyramine, ergovalin, lolitrim. Lolitrim, we're interested in having zero presence of it. Ergovalin, low levels, and pyramine and xanthitrim are 2 of the interesting ones in the context of insect protection, so insect deterrence. And what we see is that for different, in different varietal backgrounds, there are very significant differences in the level of accumulation of these bioactivities. So very strong interactions of the genotype of the plants with the genotypes, the genotype of the endophytes on key traits that we're interested in, namely the alkaloid toxin profile or the bioactivity profile in this defined symbiota with novel endophytes. This was looking at the final point of the production of the key ergoalkaloids. I showed you earlier in the context of the pangenome analysis that we can identify natural variants that have deletions in key biosynthetic genes. And for example, endophytes that will lack the two last genes, so the 2 genes encoding the last enzymes in the pathway for the production of lolitrims, LTME and J. And you see here examples, endophytes that will not produce lolitrim because they lack those 2 genes. So the question is, what happens if we have in a sense, a molecular lesion in a pathway relating to the precursors, and some of this potentially are bioactive, and some of this potentially tremogenic. So now is looking at a very comprehensive metabolome analysis, a pathway analysis for every bioactivity, and known in the case of both lolitrims, alpha-trims, and ergoalkaloids, pyramine and xanthitrims. And that picture that I showed you before from a gene presence and absence, and the picture I showed you relating to qualitative and quantitative differences in the toxin profile of the final compounds, that same more complex image appears in the context of presence or absence of all now the metabolites, the intermediate metabolites in the pathway for the biosynthesis of these major bioactivities. So now moving into what does all of that mean from a phenotype perspective, not the molecular phenotype, but at key traits. And this is just to illustrate in a couple of images. These are 2 perennial ryegrass varieties looking at the same genotype of the host without an endophyte, or with 3 different endophytes. Neotyphodium lola correspond to 3 different taxonomic groups, Neotyphodium lola, LPTG2, and LPTG3, and with 4 or 5 replicates for each. And you will see that there is a very significant difference in biomass yield and tiller number and so on, having the same host plant genome interacting with different endophyte genomes. And for every trait that a commercial grass breeding program will look at, we find significant differences from tiller number to shoot fresh weight or root fresh weight or root dry weight. Examples that this is the endophyte free plant, and that if you would inoculate it with an endophyte of this type, you would, for example, double the tiller number. If you were to inoculate it with a different endophyte, you possibly will halve the tiller number or root fresh weight. So, since that interplay and the importance of describing at the Ohm's level and the totality of the biological system that ultimately is delivered in the form of the seed on farm. And these are 2 of the most interesting fungal endophytes that we discovered using these approaches. And one is from an accession from Morocco, and the other one from an accession in Tunisia. And these endophytes produce the ideal combination of bioactivities, pyramine and lolins, and also this fungal endophytes can be deployed, so they establish stable associations with both perennial ryegrass and tall fescue. So novel endophytes in a sense for the broad deployment that have been discovered and characterized. Now, I use the example of fungal endophytes as one of the components of this symbiome, and but it goes beyond that. And and here is illustrating the diverse bacterial microbiomes that we can reveal in forage grasses. This is an example in perennial ryegrass, in which we find over 2,700 bacterial phyla in its microbiome. We can see that this metatranscriptomics allow us to find differences in bacterial species predominance in both in the shoot and the root microbiome, and in particular the root microbiome profiles seem to be descriptive of the nutritional status of the plant. And if we look more closer, we can identify as components of that bacterial microbiome in the Symbiota, revealing a range of bacterial species that particularly Aspergillum species that are known to be nitrogen fixers as well as growth phytostimulators in grasses. So that's describing some of the biology and using this Ohm technologies to characterize the biological system. Now, what does all of this mean, and how can we best utilize this knowledge in delivering increased genetic gain on farm? And so the next part that we'll describe is about integrative genomic-assisted F1 hybrid breeding of forage grass Symbiota, and specifically we'll continue with the example of perennial ryegrass. And the current paradigm that we use for breeding associations is based on breeding and selecting the grass host only. So we consider, even from a genetics and genomics perspective, the plant only, and being agnostic to other plant-associated biota. We look at few selective recombinations in a very long breeding cycle. We inoculate a single unselected endophyte. We evaluate the associations, but we never breed for the associations ab initio. We do seed generation in advance that limited heterosis, and we don't have in the marketplace today hybrid varieties, and thus limiting value capture. So what I'm trying to propose is, in a sense, that we could fundamentally rethink the way that we breed perennial ryegrass. This would apply to other forage grasses in the form of doing an ab initio breeding and selection of Symbiota rather than the grass host only, looking at more selective recombinations in a shorter breeding cycle, exploiting the broader endophyte diversity and endophyte effects beyond the simple approach. Is it the endophyte toxic to livestock, or does it confer insect protection bioactivities? Looking at a more accurate evaluation of Symbiota, exploiting heterosis and headbutt traits, and ultimately delivering hybrid varieties. And I will need to really rush So I'd like to leave you with a couple of examples of tools that we developed to deploy this new paradigm. And one is, on the one side, deploy multiple endophytes in synthetic varieties, so not only extending the concept to the grass host, but also to the endophyte, looking at ab initio, capturing the effects of the associations, and the interaction of the two genotypes of the plant and the fungal endophyte, and exploiting broader traits, reducing the generation interval and increasing selection intensity through associations, and maximizing heterosis in farmer seeds. So the one point was, how can we actually develop high throughput, large-scale endophyte inoculation methods? And we established those by using artificial seeds. This is using calcium alginate coated and embryos, and we can then deliver in that calcium alginate layer the microbiome, more specifically the fungal endophytes. We can generate approximately more than 1,000 viable Symbiota artificial seeds per FTE per day, and up to 50% of those will establish life associations. We can look at ways of selecting for stability of the association, and by using accelerated aging. This is treating, and that allow us then to, in a sense, not only select for stable association, but also ranking fungal endophytes for their broader applicability. This is treating seeds at higher relative humidity for a period of time and at a higher temperature. And you see different, 4 different fungal endophytes and 4 different volatile backgrounds, and you'll see that some of those will be very stable across germ plus pools. These are very contrasting genotypes, and others will be greatly unstable. Then the other point is being able to determine, to detect endophyte viability at a low cost and in rapid manner. So we develop a method that allows us to detect within 6 days at a cost lower than $1 per sample. So it's 5 times cheaper and 5 times faster. The previous methods that is based on a metabolomics approach. And then looking at ways in which we can increase the accuracy and reducing the cost of phenotyping in a relevant manner, and so developing then and applying capabilities in field-based phenomics. This allows us then to do automated field-based phenotype generation, assessment for vegetative biomass, persistent traits, stress-related traits, and with the diversity of optical sensors, and delivered either or deployed either with a Phenoblip or a Pheno rover or a hexacopter. So opportunities for improving then the accuracy of the phenotyping in a non-destructive manner, and as well as complementing that yield over time and herbage quality over time with a molecular phenotyping of key bioactivities. So here's looking at how can we enrich Symbiota for the optimal combination of the alkaloids that we're deploying. Setting the basis then for applying then a genomic selection, particularly in the context of refining breeding schemes, developing phenotyping tools and genotyping tools at acceptable costs. And this is the way that commercial breeding of perennial rhizos happens today, with 2 selective recombinations over 10 years. And we have now designed through computational simulations a commercial breeding program to optimize the application of genomic selection, and with good prospectivity for a doubling or tripling the rate of genetic gain. About developing hybrid varieties, the perennial rhizos, the self-incompatibility system is based on 2 loci, S and Z. So we've been able to then now map-based cloning of the genes and underpinning both of those loci from a male and a female partner perspective, developing a method for self-incompatibility allele prediction, and thus also setting the basis for a commercial F1 hybrid breeding scheme, and through bottlenecking of germ plus pools and further genotypes. Yeah. So finishing, try to give you a some examples of advances in forage systems biology from genome, transcriptome, metabolome, and phenome perspective, and that I will think the perspective of starting considering and the need to exploiting supplementary genomes and not focusing exclusively on the genome of the target host plant. There are lessons from systems biology of plant symbiomes and prospectivity to significantly improving genetic gain through integrative genomic-assisted hybrid breeding. And to finish, to acknowledging the team that has been involved in the funding bodies. Thank you very much for your attention.
Can we have one question? Yeah.
That by this integrated approach, one can increase the productivity. But my only concern will be that will it not be too complicated to control the biotas, endosymbionts, and the metabolomics, and all these things put together? Will it not make it very, very complicated? But nevertheless, this is going to be the approach for improving the crop yield.
Yeah, thank you for the question. Today in the commercial breeding programs of perennial ryegrass or tall fescue, fungal endophytes are deployed. So the technologies for endophyte inoculations in a low throughput and low cost manner are in place, and methods, seed coating technologies are in place. So I don't think that that is a burden at all, or a too disruptive step for the commercial breeding sector to adopt that. And by the way, our program of work is very much integrated with the private breeders, and so we see that these technologies could be readily adopted.
Thank you. Short time, maybe. Sorry.